Hello.
That took long enough. How did you start? How did you get involved?
In this
Out of high
business, the
school?
science,
Yeah.
yes. Well, it started in high school for me. I really enjoyed science. I knew I had an aptitude for it, but it was hard to see a career in it. I think the defining moment for me was a movie I saw in the last year of high school. It was a movie about the discovery of insulin, which really fired my imagination. So I figured that this is what science was something I wanted to do. Still didn't really have much of an idea of what it involved and how to do it, but that sort of set me on the path. So I went to university, did a science degree. Really enjoyed it. My focus was biochemistry, so that's the chemistry of life. And I really liked the biochemistry of humans rather than other sorts of biochemistries. And I suppose that was mainly because I could explain to my parents why I was doing it, that I was going to look at the biochemistry of humans, try to understand how the body works, try to understand what happens when it goes wrong, and as a result, try to come up with new diagnostics and therapies for disease. That was the grand plan. So towards the end of my science degree, I realized that, I just loved it. I mean, I loved it. I it was clear to me this is what I wanted to do for the rest of my life. Then I sort of really undertook about, you had to think carefully about how to do it, how to undertake it. It's a strange job, science. You could probably look at most jobs, you're doing one of three things. You're making something, you're selling something, or you're providing a service. Science generally isn't that. Science is really about building on a body of knowledge. And typically what happens is that you get to a tipping point where major new discoveries have made that then lead to new therapies and new understandings of human biology and other things. So it's sort of a strange job as a result. It can be quite a lonely pursuit. Is that, again, I suppose in comparison to sort of most sort of jobs, is that if you do it well, you have the gratification of that sort of an immediate response. You've done something very well. You've made a big sale. You've made something spectacular like a bridge, and you have that immediate sort of feedback. With science, that doesn't happen so often. Most of us, what we do is we build on a body of knowledge. Occasionally, we get very lucky being in the right place at the right time and thinking about the right thing at the right time, and you can make big breakthroughs. But they take, generally, particularly in biological science, it takes a long time for them to be recognized and appreciated. And so not having that sort of immediate gratification in the work that you do, it doesn't work for everybody. I've seen exceptionally talented people over the years where science just isn't for them. It's not for them. Their whole process didn't work for them. But it really worked for me, and I just loved it. And I still love it. And it's hard not to love, because at the end of the day, what it is, it's discovery. My job is to discover things about, us. How does our body work? What happens when it goes wrong? That's okay.
No, it's not.
It's okay. Was it something I said?
I thought you were really good. Yeah, okay, Mr. Arnold? No. I think no. Okay.
Okay, keep going. All right, so
Where were we?
yeah, what is it about the nature of the job? So at the core of it, it's about discovery. It really is. That's what it's about. Our job is to discover new things about the human body, how it works, and what happens when it doesn't work, when it goes wrong. So how could you not like that? I mean, it is enormously challenging, and mainly because our body, living things are enormously complex on a scale that's really hard to imagine. So what we're trying to do is understand how that enormous complexity works. And if you throw a spanner in the works, what happens? Just to give you some idea, if we look at our level of understanding of human biology, and let's say 10 is when we understand everything, I would think we're probably about level 3 or 4 currently. So there's so much we don't understand. And because we don't understand it, it's very hard for us to make, really real breakthroughs that I suppose that, have general applicability, in the population because of that complexity. Let me try to explain that in a different way. Is that, okay, if we look at our big diseases in society, cardiovascular disease, cancer, obesity, those sorts of big problems in society, these are what they're known as, they're multifactorial, meaning there's many things that influence these diseases. And each individual, how they manage these differences, it is different. So it's very hard to say that, okay, look, we understand this, how this is working in this situation, but more often than not, it doesn't apply across the board. So it's that huge complexity that makes, the big breakthroughs rare, but they happen. And it also often, I find it remarkable that they do. I sometimes find it remarkable how successful we've been. Let me give you an idea. So 100 years ago, our average life expectancy was half what it is now. I mean, we were living till 45, 50, if we're lucky. Our life expectancy now is in the high 80s. And so that's 100 years, and that's almost entirely due to health and medical research. So it's working. The thing about it is we can rarely predict where the next, almost never predict where the next big breakthrough is. But as an enterprise, it is working. It's not an enterprise you can stage manage. The way it works is that, you try to find, talented individuals who are dedicated in terms of what they're doing. Just give them the capacity to do what they want to do, and great things arise. It's not a profession, it's not a sort of a game where it works by committee. It never has, and I don't think it ever will. And as a result, it's a bit hard to explain, the nature of it, because it works that way. It really comes, you've got curious individuals trying to discover something. And they think, okay, this is a big problem. I'm going to try to figure this out. And so they'll start here and they think, well, okay, I'll do this, and then I'll do that, and then eventually I'll get to my answer. What almost always happens is you do this, you get here, but you're not getting the answer you want, which takes you in a different direction over here, which then sort of puts you down a different path, and that path might change again. So you might have started off with the idea, I'm going to solve this problem over here, but you end up solving, a couple of different problems along the way, which actually may be bigger problems than the one you started out with. So it's as a result, you just can't stage manage it. You do good, careful work. You make sure you're, look at the results and just try to open your mind to what they mean, and then try to make good decisions as a result. I think the thing that haunts me about the game is that, how many observations, how many experiments I might have done, or my team's done over the years, that have given us results that we really don't understand. That could have resulted in a major breakthrough, but I just wasn't clever enough to see it. And occasionally that sort of haunts me. But that's just the nature, it's the nature of it, and it's just hard not to love. How could you not love it, really? It is a privilege to do it. It's extremely competitive business, but it needs to be, because at the end of the day, it's quite expensive. Most of the research that we do is funded through the public purse. So we have to spend it wisely, and we have to try to spend it efficiently. And as a result, it's extremely competitive. To give you an idea, if I had applied for a grant to sort of do something, to try to solve a problem, the success rate of that in that granting scheme might be, say, 10 to 20%. So they, might only fund 1 in 10 or 1 in 5 applications they receive. So you need to be in that top 10% more often than not to sort of sustain a career in the business. Yeah.
That'll be clear in a sec. Is that a role of distant funder?
Okay.
I think it was a trolley up there.
It was, yeah.
It's good stuff.
Is this what you're after,
Yeah,
that sort of stuff? Okay.
Content.
Okay.
Then we'll move into
Okay.
state cancer. Okay. We'll continue as we were.
Okay.
Just recap that a little.
All right. Yeah, it is just in terms of the nature of the business, it's also very competitive. As I said, it sort of needs to be because it's an expensive business. It's also a business where it's easy to be wrong. You know, I say, look, I have this idea about how this works. I'll need $500,000 to sort of see if I'm right or not, but you can often be wrong. Okay. You can't be wrong all the time because then you won't get funding for your next big idea.
Is being wrong a bad thing?
No, and it's not a bad thing because, as I mentioned, typically what you find is unexpected things. And the big discoveries throughout, human history, particularly when it comes to human health, they were serendipitous. They were, look, we got a result we didn't understand. But then I thought, well, it might mean this. And it so happened that it did. And that leads to, great things as a result.
Do you have some favourite examples of that one? Or those? Serendipitous?
Yeah, well, there's a great, just in terms of, say, okay. Good serendipity.
Probably penicillin, but
Yeah, that's a famous discovery. Well, yeah, because it was unexpected. Probably the discovery of the electron back in the late 1800s that led to X-rays and MRIs and medical imaging, all because of the discovery of the fundamental properties of the electron. When that was first made, it was a fascinating but physical. It was in the world of physics and engineering, where it had enormous application. Obviously, it led to the electric motor, etc. But it also led to sort of major breakthroughs in health and medical research. All the sort of imaging that we take for granted now that revolutionized our diagnosis and treatment of disease. And so when it was first discovered, the nature of the electron was really never even conceived that it would lead to these sorts of breakthroughs. So I think that's a classic example where you discover something in one field for completely unrelated reasons, and it has applications in others.
Well, it's like the space program. How many things came out of the space program?
Yes.
Not least the microwave oven.
Yes.
No one was expecting.
Yes.
And Velcro.
Velcro.
I thought Velcro was a Swiss naturalist, actually, taking burrs out of his socks.
Okay.
I know these things.
So that's the nature of it, and I think that's and as I mentioned before, you just sort of can't stage manage it. It's unpredictable where these big discoveries are going to come from, and so it's sort of a profession that. It's a profession, I suppose, that we're in it, are constantly justifying, the expense. That, look, we're asking the government to sort of support the work that we do. Most of our research comes from government. And, we can't hand on heart say, look, if you give us these funds, this is what we're going to discover, or this is what we'll do. We can't say that. But we're forced to say that at some level. So it's a constant justification, which again is not a bad idea. We complain about all the grant writing we have to do, but some of my best ideas have come through grant writing. It forces you to read the literature, really forces you to think, deeply about the problem. And often, I realize that, this is interesting. Didn't realize that connection. And, these things are good. So it's all part of the mix. And we all do it differently. So when it comes to science, as scientists, we all approach it differently. The way we think about it, the sorts of experiments we do, how we manage our team, we all do it differently, which is also part of the really enjoyable aspects of what we do.
Are some jealous and others not in their nature, or collaborative or not?
Yes, yeah, it's the same. You find the same human failings, I suppose, the human peccadillos, if you like, in research scientists as you do in every other profession. We tend to probably have a higher proportion of strange character traits than others, and that's perhaps the nature of it. We're often not great communicators. We're often quite happy to be, in our own little worlds, thinking about our own problems. So it tends to sort of attract a certain personality type as well. I think there's also generally also, if you decide to do this as a career, there's a sort of a risk-taking element too. You need to be happy with the risk. You need to be happy with that. Look, I could be wrong. I could be out of grant funds in 2 years' time, and I could be looking for another job. So you sort of have to be comfortable with that uncertainty of the whole thing. But I think those of us who love it sort of almost, we like that uncertainty. We like coming to work really not knowing what we're going to go home with. We sort of thrive on that unpredictability. So yeah, again, that's sort of the nature of the game as well. It doesn't work for everybody.
But so you went into science.
Yes.
When you went into science, that's not necessarily research.
No, it's not. No, you do science. You basically science. That's what you're doing. You're learning about science. You're learning about physics, chemistry, mathematics, biology. It's understanding. It's learning about what we currently know. Knowing that, and this has been said before, knowing that in 20 years' time, half of what we've learned is probably wrong. But yeah, so you learn, you from that knowledge, you'll be drawn to it or not. You'll be fascinated by the whole process, which I was. I was just fascinated by the complexity, of how we're made and the endless stuff that we didn't know. So that fascinated me. But so you did the science degree. Then I chose to do a research degree. So it was a PhD, doctor, which is a research degree. So in a PhD, what you do is you learn how to do research. Okay, you're given a problem. You learn how to design experiments. You learn how to interpret the data, and you learn how to solve problems. So it's very much research training. Then following your PhD, if this is the track you want to go down, that's a full-time, sort of researcher for the rest of your career. The next stage in training is a 4 to 6 year period, which they call postdoctoral research work. And this is when you tackle bigger problems. You're given more independence in what you do. You still, have supervision from the senior people in the lab. You're still reliant on them typically to provide the funds for the work you're doing. And it's at this period where you really decide, do I have what it takes? Is this what I want to do for the rest of my life? So when you get through that period, you need to be successful because it sets yourself up for that next stage in your career, which is that time where you try to be independent. You try to become an independent investigator. You try to have good ideas, you try to write good grants, you try to get your first grant, you try to employ your first researcher, then you have the beginnings of a lab. And then you build that lab with more research funding, with more research, more success, better ideas, and then you try to sustain a career in the business by doing that. It is a job that often, it's interesting if you look at the numbers. There's not many people that will sustain a full-time research career over the course of their career. I suppose it's like most professions. You'll go through a period in life where you're most productive, I suppose, and also your ways of thinking are optimal. They're less set. They're less. I'm trying to think of the word. There's they're malleable. Yes, that's right. And so often you'll find researchers like they might have a 10, 15, 20 year period where they're highly productive and then it'll drop off. Some bloom late, some bloom early, particularly I suppose in the hard science, mathematics and physics. The saying is that if you haven't made your major discovery by 30, you're probably not going to do it. And I think that relates to that sort of malleability of thinking, not being constrained by the norms and the general impressions about how things work. In biology, it tends to be a bit longer because it just takes a long time. If you have an idea, you think it's a big one, and you know that it's right, it can take a long time to actually prove that it's right, and for there to be practical benefits at the end of it. It can take 10, 20, 30 years sometime. That's why in biological science, if you look at the Nobel prizes that are won in medicine, for example, it's often discoveries that were made 30 years prior. So that's the nature of the game, is just, you know, trying to sustain that intensity and that you know, the love for what you're doing, and that over that course.
What are some of your ideas?
Some of my ideas.
Steve?
Okay. All right. So look, there's I suppose there's two. One's more of a general one, but then I can lead it into the cancer if you like.
Yes.
Yeah.
Sure. Absolutely.
All right.
Yeah. If it wants to think up, it's running now. No, it's
Talking about ingredients for research, weren't we?
Yeah, it was good for research, and also what your thoughts were, what led you, and you were going to
Yes.
go to.
Okay. Sure. Okay. So I suppose that probably in general terms, the biggest thing we've done, or the most important thing we've done, is about how proteins work. So you've got your genes in your cells. You've heard of your DNA. Your DNA is like the blueprint of your cell. It encodes for the proteins that are made by the cell, and the proteins are the machinery of life. They do all the stuff. Okay. So you've got DNA encodes for all the proteins. It's like the blueprint. Their job is to make proteins, and the proteins do everything in the cell. So we discovered this aspect of how proteins work, which was unexpected. You know, it was thought that this aspect of proteins was, you know, set in stone. They didn't do this. Through, you know, again, it was an example. We had an observation. We're working on a protein. It was doing something odd, which we couldn't understand. So then we thought, okay, let me describe. I'll let that a bit more explanation. So proteins are held together by two types of strong bonds. They're called covalent bonds. You've got the bonds that link your amino acid building blocks, and then you've got other bonds that cross-link the polypeptide backbone, so to speak, called the disulfide bond. Now, these disulfide bonds, they're in most human proteins. They're quite common, and they were thought to be sort of inert motifs. They were thought to act like struts in a building, if you like. They're thought to sort of hold the protein together, help it get through its life. We were working on a protein that was doing something odd, and for some time we couldn't figure out what the situation was. And then we discovered that what was happening is that it was one of these disulfide bonds in the protein that was being broken. Okay. Prior to this, it was thought that these disulfide bonds were inert. You know, they were strong bonds. Once they formed, that was the end of it. But we found that one of these bonds was being broken in the protein and then controlling how the protein works. Okay, so that was the surprise. And so then it wasn't much of a stretch to think, well, if it's happening in this protein, it could be happening in others. And that turned out to be the case. And there's a whole subset of these bonds that control protein function, when they're either broken or they then, in some cases, when they form. And so that's motivated our biology, trying to understand the significance of this biology, and, you know, how we can use this information to sort of, you know, understand disease. And this is where cancer comes into it. So one of the things that we did was make small molecules. We wanted to try to understand this protein chemistry. There wasn't any tools available to do it. So we made some tools to try to understand these funny disulfide bonds. One of the molecules that we made did something unexpected. What it was doing was concentrating in the dead and dying cells in our population. Okay, so cells are like human beings, I suppose you like. You know, if in a given population, there'll always be some of us that are, you know, that are dying, or some of us that have passed away. Cells are the same thing. What this molecule was doing is it was picking out these cells. So we thought that we could use this property of this molecule to maybe look at the dead and dying cells in tumors. Okay, so our initial interest was that, okay, so we know that cancer therapies, their job is to kill tumor cells. We thought, well, maybe we can use this molecule to measure that in tumors. And it turned out that we could. And there was really no way to do this, you know, prior to this approach. And so the other thing, where this is leading now is this big problem in cancer known as residual disease. So it's really probably the most intractable problem that we currently have in the disease. So let me tell me what it is. So there are two types of cancers. You've got your blood-borne cancers, your leukemias. They constitute around about 5 to 10% of all cancers. The other cancers are the solid tumours, like you have, breast, prostate, brain, lung tumours. These are the solid tumours, and they constitute, they make up the other 90% of tumours. Now, when we try to treat a tumour, in most cases, we're pretty good at doing it. But the problem is we don't kill it all. So we'll have a therapy that might kill 90% of the tumour, but there's nearly always, a fraction of the tumour that just won't be killed by the therapy for a couple of reasons. First of all, there's a population of cells in the tumours, or tumour cells, that just aren't being killed. They're refractive. They're not sensitive to the drug that you're giving the tumour. So they're not being killed. So they will survive. In some cases, the process of treating the tumour results in a small population that then becomes resistant as a result of the treatment. So this problem of residual disease. And why it's a problem is that you might take out 90% of the tumour, that 10% remains will then regrow. And because it was already resistant to the therapy that you threw at it to start with, it's going to be resistant to it the next time. And so this is the problem we have with, big problem we have with cancer therapies now, is that they generally become less and less effective each time you use them. And that's because you end up treating an increasingly resistant tumour. So this is this problem of residual disease. So what we're trying to do is get around this problem. We're trying to develop a therapy that's designed to become more and more effective with each administration. And it's based on that serendipitous observation that we made when we're trying to understand a fundamental aspect of how proteins work. So this is a classic example of how discoveries in science typically arise. We made this molecule for one reason, started working with it, found it was doing something odd. Then we invested time and effort to understand what was happening. Then we realized there were some practical implications of this compound. So then we basically followed our nose, to where we are now. And so we can, so that's really our a major focus with this technology now is trying to get around this problem, or trying to tackle this problem of residual disease. If we can tackle that one, that'll make a big difference.
How are you going with that?
Okay, so it's okay in terms of how things develop. So we discovered this compound. We figured out how it works. We showed that we can use it to sort of look at dead and dying tumour cells in animal studies. We showed that we could use it to treat tumours by tagging this molecule with something that will then kill the tumour. So we tag it with something, it finds its way to the dead and dying tumour cells and kills all the living ones around it. Then we realized that, okay, because we've done that, the next time we administer this compound, there's more target for it, because there's more dead and dying cells from the first time you administered it, okay? And so with each time we administer it, we're creating more target for the next time. And so we have this sort of self-amplifying sort of cell kill that's going on. And so we showed that in preclinical studies, and now we're into our second clinical trial with the development of this compound. So what the future holds really is really very much clinical development now, and that look, we're probably looking at a 5 to 10 year time frame. I think before we're at a point where we can say, okay, this is ready to be used. This is we're going to go to the regulatory authorities and say, this is our data. We think it'll be useful. It'll help cancer patients. So that's where we are now.
How many clinical trials are there typically?
It really all depends on the technology. So generally speaking, the first trial is just a safety one. You've got to make sure this molecule is not going to do more harm than good.
So yeah, that doesn't hurt.
No.
We're
So
not
we know
worried about what it's going to do.
we know that's the case. We know it doesn't hurt. It doesn't have any toxicity. Any off-target effects or side effects. The second trial is does it work? So we're into those trials now, and we know it works. What we're going to learn from this trial is that what's our first indication? What is the first cancer type that we should investigate this molecule as both a diagnostic and a therapy? And that's what these phase 2 trials will tell us. Should we be looking at pancreatic cancer? Should we be looking at brain cancer, colorectal cancer, what would be our first indication? And that's an important one because we sort of need to get that right. You know, if we're successful with that first indication, if we've got a molecule, a therapy that works, and it's better than the existing standard of care in that cancer, well, then I think it's all downhill from there. And so that's a very key decision, and that's a decision we'll be making in the next year or two, and then we'll progress with that trial, and fingers crossed, we'll get through it in flying. Yes, that's the hope. But we also look. There's also so much uncertainty still. We may discover something that that was again totally unexpected. A side effect or an off-target thing that could be difficult to get around. I don't think we will, but there's still uncertainty. There's uncertainty through the whole process. So really our job now is to just try to make the best decisions we can. Make the most informed decisions. Talk to the smartest people. Get their input. Is this where we should be going with the development of this molecule or not? And if we get that right, I think we'll be okay.
Is that uncertainty part of the appeal?
Yeah, it is, but at this stage, it's just more scary. It's at this stage, the uncertainty is you get to this point with the development of the technology. It's been an enormous investment already. Both in people, power, money, everything. Now it's about really just trying to make the right decisions. If we make the wrong decision, it could put us back 5 to 10 years, in terms of where we go with the development of it. We have to get that right. And there's also outside factors that will influence all these, how we make the development of the compound. There may be competing technologies that come along to do a similar sort of thing or the same sort of thing that might be better. That's not the situation currently, but we don't know whether that's going to be the situation in 2 years' time. So there's these things you can't control. And so that sort of uncertainty I can do without. The uncertainty in terms of...
And yet?
But look, that's what my job is. That's what my career is. That's, it may be terrific and no one will be happier than me. It may fail. That's the brain, that's the possibilities I signed up for. That's part of the job. Just got to suck it up, move forward.
So what's the connection with the ACRF?
Okay, so in terms of develop.
Be nice.
Okay, well, no. So look, others will tell you this too. ACRF, Australian Cancer Research Foundation they provide a unique capacity for medical research, cancer research in this country. They provide the technology to enable us to do the research that we need to do. And so I've been fortunate to over the last 15 years to win 3 grants from the Australian Cancer Research Foundation, and all 3 of them were very important, if not critical, for the development of the technology I just talked about. So ACRF provided the funds to us for us to purchase the equipment that we needed to develop this sort of therapy approach that we're working on. And so, absolutely key. I can hand on heart say that, without their support, I don't know how far behind we would be currently. They provide this unique funding, window that we all absolutely depend on in this country as cancer researchers.
Should there be more philanthropic?
Yeah. Oh, look, there's no doubt. So just to give you some idea of, I suppose in the profession of how it works, typically about 70% of the funding that we, that we win comes from, say, federal or state government. About 30% comes from, organisations like the Australian Cancer Research Foundation and other philanthropy and other organisations like cancer councils and such. And so, we're so dependent, that 70% on federal and state government, and that changes all the time. So, the nature of the research that both federal and state governments fund, it changes all the time, from year to year in cases. So some years there's, the sorts of things they want to fund doesn't work for what you're doing at all, And that's when we particularly rely on the other sources of funding, that 30%. But it would be terrific if we could get it to parity. You know, if we could, you know, if it was 50% federal state government, 50% philanthropy and foundations like the Australian Cancer Research Foundation, we would be in a much better situation.
What's generally the quid pro quo between government and philanthropy? There is an agenda, sometimes not, sometimes.
Well, it depends. No, generally, some cases, you know, philanthropic funding is tied to a specific problem. They want a specific outcome. And for some that works very well, for others it doesn't. A lot of philanthropic funding, though, is, again, it's funding curiosity-driven research. They're not being prescriptive about what you can work on. They recognise you've done good work in the past and they want you to take this money and do good work with it in the future. And for researchers, you know, I suppose like me, the fundamental discovery researchers, that's the sort of funding that's dear to our heart, you know, because it gives us that flexibility to change directions if we find something more interesting to do. So in terms of the quid pro quo, there's occasionally that you find that there's more industry funding where there's, you know, where it tends to be very applied, you know. And I suppose many of us are relying more and more on industry funding to sort of help fund, you know, our laboratories' work in general. But it, you know, it doesn't work for everybody because it's very prescriptive. They want you to, you know, you've got maybe a unique model. They want you to test their drugs in your model and provide them with the data. And that may help you sort of support and fund the model in your lab, but it's sort of not necessarily the work that you're driven to do. Okay. But in this country, it's still a minor part of our funding. Industry funding, you tend to find more of it abroad, in the US or Europe, less and less so here.
Because that's the passage. You're supposed to make the breakthrough, find the breakthrough, I guess, to allow them to make the drug, to allow them to make a profit, but they have to put the money in to make the profit.
They do. They do, I suppose, you know, in terms of that aspect of things. So it's probably, Australian medical research is really well recognised for its quality. You know, I think our fundamental discovery research in particular has always been very strong, and it ranks with the best in the world. What we've failed to do currently is take some of the discoveries that we've made in the bench and turn them into industries. Okay? And there's plenty of reasons for this, but we haven't done that yet. Now, it's not because of our size. Let me give you an idea. So if you look at, say, the populations of Israel, Denmark, Sweden, who combined population is not ours, they all have, you know, pharmaceutical companies in the top 20 in the world who employ 50, 100, 150,000 people, you know. The most valuable company in Europe is a Danish company currently, known as Novo Nordisk. So the economic benefits that a major pharma brings to a country are huge, and we haven't achieved that yet. For a lot of, we've made breakthroughs, but unfortunately they've all been developed overseas, you know, for a variety of reasons. I think to solve this problem to a large extent, look, many reasons, some bad luck, there's no question there's been some bad luck along the way. Just the timing wasn't right for the discoveries that we've made. But our biggest problem is venture capital. So the way it works is if you make a discovery in the lab, you think you can turn it into a diagnostic of new therapy, typically you go to venture capital and you say, look, you know, I need $10 million to get this technology to a point where I can, you know, then interest big biotech companies or major pharma in developing it for the clinic. Okay, so, and to give you an idea, if we look at all the venture capital for biotech in this country, it wouldn't measure up to, say, a mid-sized venture capital firm coming out of California. You know, so we just don't have enough biotech venture capital in this country to sort of develop the discoveries that we make. that's our current. That's why I think it's a big part of why we haven't. Why most of our major discoveries have gone overseas is that because we couldn't get that funding to develop them here, so we had to get that find that money offshore, and then as it developed, then that money remained offshore and was developed overseas. Okay, so we need to get the development to a point in Australia where then it can lead to a major pharma, a major industry, and we just need a lot more biotech venture capital to do that. The hit rate is small, but now this is speculating, but it's interesting. You may not want to use this. I don't know how factual it is, but just to give you some idea.
Well, we'll run it past.
Yeah. Okay. So, people say about biotech is that the hit rate's so small. I mean, if you got something successful, I mean, it's they're $1 billion a year drugs or therapies. So. If you might discover something in the lab, and then to get that to a point where it works in living animals, might be 1 in 10. To take that discovery to something that works in humans, it might be 1 in 10. So from that discovery in the lab to something that works in humans is about 1 in 100. And I know those numbers are high, but apparently it's not too different in the mining industry. I've been told, this is where a lot of our venture capital in this country goes into primary industry and mining. And speaking to people involved in that industry, they'll tell you that, to find a new deposit, the odds are probably 1 in 10. And then to be able to extract that ore or whatever it is to make in an economical sense is probably 1 in 10 too. So again, the odds there are probably not too different than they are in biotech. It's just that there is that perception, I think, in the Australian venture industry, whereas it's high risk, too high risk, but I don't think it necessarily is. But look, the bottom line is we just don't have enough money in that space and people. In terms of developing, new diagnostics and therapies to the clinic, it's a real specialty. You need experts that know what they're doing, that have done it before, made mistakes before, fixed them and moved on. So we don't have that. We don't have that enough of that expertise either. Money's one element of it, and it's probably not the most important. It's people. It's the right people with the right expertise is even more important. You can have a ton of money, but if you don't have the people making the right decisions, it's not going to happen.
How would you attract the right people, and what would you warn them?
Well, again, it's just having a bigger venture capital industry. If our bright science or med students see this sort of biotech universe as a really an attractive career option, more of them will go into it. If they can see, if they have they have that sort of entrepreneurial bent, they have that science background, there's great opportunities for them in biotech, as venture capitalists or venture partners or whatever whatever they are. And we just need more of them. And the more success that you see in that industry, our smart young Australians will migrate to it. We just haven't reached that tipping point yet. There are more and more coming into the game or that profession, but not enough. And one day we will have a major farm in, the top 20 in the world that will employ 100,000 Australians. We will. It's just that I don't know when.
Anything you'd warn people about going into the realm of research, especially bioresearch?
Warn them about? Well, in terms of when I talk to, my own team and the people I mentor, I think you need to have your eyes open really early on. You need to know there's different stages in your career. You've got your degree, you do an honours degree, you do a PhD. That's really research training. More formal research training is your postdoctoral period. So there are periods in your career where you need to be successful at each stage. Okay, you need to make good decisions about where you do your PhD research, for example, who you choose as a supervisor. So you need to find environments at each stage in your career where you can be as successful as you possibly can be. And a lot of, and see, it's the thing about this game, it's sort of easy to drift. You say, well, I'm going to go with that person because I like him or her. That's not a good decision. You know, you've got to make decisions on where you work based on the quality of the team. Is that a team where will provide you all the capacity you need to do what you do? And because you can't, most of us, you typically can afford one bad choice in a career. You can usually recover from one, typically not two. Because it's so competitive, you can't have 2 or 3 years of no productivity because you won't then be competitive with your peers when it comes to grant funding. So a lot of it's about just, being smart about how you manage your career, knowing what you need to achieve at each stage. If things aren't working, make a change.
What was your course? Who did you pick?
Okay, so I did my undergraduate and postgraduate at the University of Queensland. I was very happy with my PhD supervisor. I wasn't so fussed about what I was doing. I just knew that people that have worked in that lab have been very productive and have gone on to do big things. So I thought, okay, this is a good environment, and it wasn't a mistake. I really enjoyed my time in that lab. I got great mentorship. Then I had to choose what's known your postdoctoral period. So I moved to the States to do a period of postdoctoral work. If I had my time again, I don't know. I should say, if I had my time again, I'd probably make a different choice. But then again, I think I made a wrong choice. But then about 6 months in, I realized that look, I made a bad choice. I just got to make this work, And so I tried very hard to make it work, and ultimately it was successful. But in hindsight, I probably would have made a different choice. I was just a bit naive. I went to a lab that wasn't on the up. They'd done some good things 5 years previously, but they were sort of,
East Coast or West Coast?
It was Midwest. But they'll know who this is, so bleep that. So I've just been...
There's no point.
Okay, that's fine. So, and after that, I went to a second postdoc in Sweden, and that was a terrific experience. And then when I came back to Australia to establish my independent research career, I had, there was a guy that...
You're here, we're lit.
Okay,
Having fun.
just to one thing, if I tell my people why I do this. Okay, so, the really appealing thing about this job is that you're very much your own boss. You can do what you want, when you want, how you want. You have to do it well. You have to produce good things at the end of the day because you'll soon run out of money and people won't fund you anymore. But it's a real appeal of the job. You're very much your own boss. You're in total, direction of your career. So, and I find that a very appealing thing. You'll live or die on your own decisions. You've got to be comfortable with that. But, it's very appealing. The other thing you need to look at, is good mentorship. If you're wanting to get into this game, is really good mentorship. You'd find good labs. You need senior people to sort of support you, advise you, help you manage, the local environment in terms of the politics and everything else that goes with it, which is like any profession. Good mentorship is key. It doesn't matter what stage of your career, mentorship is important. I still have mentors now that I trust enormously. So that's the other thing. Don't look for good mentors. That'll make a big difference.
My boy, it's fantastic.
Is that okay?
I think so.
Yeah. All right. Good.
Is there anything else you want to say? Do you want to go home now?
No, unless you want to hear any other drivel.
Wasn't drivel. I think you were very succinct.
Okay. Good.
No, I think that you can ask a question. but-
Yep.
Just addressing Stu, if you could discover anything, something you've been working on for the last 20 years, or something that's been in the back of your mind that you really like to understand, or what would it be?
If I could discover something new. Yeah, so, okay. What we've discovered recently is that, you know, it again relates to these disulfide bonds in proteins that we were talking about. What we've discovered recently is that it was thought that you have a gene, the gene makes a protein, and there's one form of the protein. What we've found now is that in many cases there's not one form of the protein. There could be hundreds, if not thousands of forms of these proteins. For reasons I won't go into, bore you with the details, but so that generally what we found is that's the case. So that for some reason we've evolved to create really, populations of proteins, and this is important for how they work. I'd love to figure out why that is, and how that is controlled. How could we possibly take a single gene and end up making, in some cases, hundreds of thousands of versions of that same protein? And this is important for how they work. And why is this? Wouldn't you just want one form of the protein to do one thing? But no, it's not like that at all. So there's a sophistication about how these proteins work that we're just beginning to understand.
There's the same proteins in your lips and your ears and your nose.
It probably implies, in generally. We're finding it's probably the norm, not the exception. That's what we're finding. And it was a total surprise. Again, it was based on, the technology that we're using was funded by the Australian Cancer Research Foundation. We used this to investigate something, and then we found there's something odd was going on, the usual sort of thing. Then we discovered this thing about these proteins, and so there's a whole level of control that we're just beginning to try to understand. And if we can understand that, I think that'll lead to sort of lots of insights about, why some people respond better to some drugs than others, why some people don't respond at all, why some drugs work in others, some people, but not in others. Why do they work initially but then stop working? It may relate to this, this aspect of these proteins that we're working on, this aspect of protein biology that we're working on. So I think that's where the big discoveries are. And so that's what we're working on currently.
That's pretty inspiring.
Oh, well, I like it anyhow.
No, I actually mean it.
I think.
Okay, I think we're done.
All right.
Thank you for your time.
Good. My pleasure. Thanks, guys. Good luck with the rest of it.
Thank you, mate.